and I think we're a pretty small group over here, so we can be very informal in terms of conversations. And I think this data challenge, I hope there are many more such challenges, and there's excitement in our community, because the potential of what impact we can have from some things like this is humongous. So, but Tushya, why don't I hand it over to you right now?
So welcome everyone. Thanks for joining us today. So this is Tuğçe. I'm Associate Professor in Eindhoven University. And together with me, we have two colleagues from Merck, Bob and Anthony. So I'm really grateful that they could come today and then answer any questions you might have. So I'm not sure how you would like to handle Bob, Anthony, but I propose maybe we can just start the discussion, and then, people can ask questions. So let's just have an informal chat about the data.
Yeah, and maybe just as we're getting started, Anthony and I can introduce ourselves just a little bit more. So I just want to call out, so I'm Bob Meyer. I work in at Merck Co. Inc. from Rahway, New Jersey, here in the United States. So I'm based out of suburban Philadelphia. Outside of the U.S., we're known as MSD. So for avoid name confusion with the counterpart company. I'm a senior principal scientist at Merck & Company Inc. And I work on new technology advancement and are really to bring new products forward into manufacturing. So I primarily work in oral solid dosage manufacturing, tablets and capsules, that kind of thing. But I'm interested in all types of new technology, and of course how just how data can transform how we do our work, right? We're kind of moving from a point in the pharmaceutical industry where we had very limited data sets. You might have like 10 measurements over the course of a million tablet batch, and of course this publication we had 300 million different measurements over the course of a batch. So it's just a tremendous transformation. And so what was it, 7 or 8 years ago, my colleague Anthony Tantuccio, he was at Merck & Company Inc. with me at the time. He's now at Hovione Inc. doing very similar research, but we endeavored to do a production batch bigger than we'd ever done before, and it's small relative to a lot of other industries, but it was big for pharma, and so we were excited to share the data. So that's my job. Maybe Anthony, if you want to introduce yourself really quick and where you are these days.
Yeah, I think you did a good job. Let them know. I've been with Hovione, a smaller company, a CDMO, contract development manufacturing organization, and basically we manufacture for larger companies and also develop new products in all stages of the product life cycle. So I moved to Hovione into a role as a fellow scientist focusing on continuous tableting, or continuous manufacturing for all solid dosages. Much a continuation of some of the pioneering work I was doing at Merck with these guys. And so Hovione is a specialty CDMO, and we're really focused on bringing continuous tableting to the masses. And it's still, the wealth of data that comes from all these machines is still, it's still a lot to manage and build. And I was saying earlier, just by having the data doesn't mean it's valuable, right? I think the big challenge is how do we get all this data and extract real value out of it and make real step changes in the way we work day to day, right? And really optimize our workflows and minimize rework. And there's so many things we could do if we just could tame the data, right? So I hope that this effort will help spur a lot of that innovation, and I'm happy to participate and be here today.
All right. Yeah. Thanks, Tucci, for giving us a minute to do that. So.
Sounds great. So shall we start with the question? Feel free to just,
Tucci, maybe you should tell people that you are, you are at Eindhoven, but I'll let you, and Eindhoven, go ahead. Give a couple of sentences about yourself.
Yes,
too.
I also have a job update. In January, I'm moving to Boston to join Northeastern University IE department as associate professor with tenure. So it's a quick update from me. Looking forward to that.
Yes, Fahim, she's in our neighborhood after this, right? It's wonderful. Okay, so go ahead. Anybody with questions? Sounds like Tucci's open.
Very basic questions. Is the exact number of machines in the same as what is described in the case, in the paper that was provided with the, meaning the number of feeders, the number of blenders, is it exactly the same, or is that the diagram a stylized diagram as opposed to actual?
No, yeah, it is exact. It shows, I mean, there's some, in terms of the system overview, maybe I'll just describe briefly, and we could even share the screen and go over the diagram if you want, but it's,
Yeah.
there's 6 feeders that, feed into 2 continuous blenders that feed into a tablet press is the essence of it. There's, depending on which blender the feeder is feeding into, we will give it a different name. So you'll notice that one of them is labeled as feeder 7. It's the feeder in position number 7, meaning it's going into the second tablet press. But so there are 6 feeders, just like there are 6 cartoon, 6 in the diagram. But there are actually 8 different positions of where they could feed. Basically, a couple of them will pivot in place to because they could optionally feed the first blender or the second blender. Did that make sense?
Kind of.
I know.
Maybe it's better to show the picture, and then
Sure.
I'm sure others have the same question because it's very basic.
All right, let me, I'll try sharing my screen, and hopefully I don't show anything too crazy.
But it's interesting, while Bob pulls it up, that you mentioned machines, right? We consider this
Yeah.
one actual machine,
Oh,
right?
the
It
whole
just
thing is one
has
machine.
many,
Okay.
it has many modules inside of it, which some of them operate on their own independent controls, and others are coordinated by a higher level supervisory control and data
Okay.
acquisition system.
Yeah. I was a mechanical engineer, not a chemical one. So
Yeah.
still thinking machines, not.
That's me too, actually. I don't have a chemical engineer, but a lot of this is mechanical in nature.
All right, so this is the diagram. Hopefully that's big enough to see.
Yeah. Yeah.
So I was just referencing, there's what's called loss and weight feeder dash PD1, PD2, 3, 4, 5, and then it jumps over to number 7. So there are 6 boxes representing 6 feeders. But this one is configured for position number 7, was all I was saying.
Okay. OPD is position 7. So is the output of Blender 1 going into Blender 2, or is one
Yes.
sometimes empty and no? So there is stuff going always into Blender 1, then into Blender 2.
Correct.
Like in the picture.
Yeah, just to go over the diagram again, these 5 loss and weight feeders all feed into Blender 1. There's a continuous flow into that blender, and there's a continuous flow out of that blender at the same rate. So roughly 99% of the total mass flow rate it goes, I guess, all of what's in Blender 1 goes into Blender 2, and then about 1% of the total mass flow rate also gets added this lubricant from position 7, and that completes it, and then all of that flows into the tablet press continuously. So equal rates going in and coming out at all times.
Yeah. So is there I think it's called residence time in Blender 1, Blender 2, because it'll take some time, but obviously the flow in is equal to flow out. So there must be sort of inventory or a resident time in Blender 1 or Blender 2. So what is that known? I mean,
yes, we measured that at a number of times across our campaign, and there was a companion paper in the references Anthony and I published a few years earlier that published details of that residence time distribution.
Okay.
But I do believe the original paper, 120 hours, does have the residence time for this data set in it.
Yeah, that's what I'm saying.
Yeah. Okay.
Okay.
I thought you meant
and I'll just scroll to the bottom and point that out briefly if you want to find it. Sorry. But one of the questions we had asked was, we think that the data set that we provided here would offer different ways to measure that residence time distribution based on the the signal data there. And so we were, kind of challenging the community to come up with their own measurements and compare them to what we had come up with and see if there might be advantages in terms of, signal to noise or just ease of measurement, that kind of thing.
Yeah.
Okay. So if you take your...
Oh, this paper. Okay. Yeah.
Yeah, this one. Holman et al.
Okay.
So that was our original paper that describes in further detail the experimental work, and you'll see lots of RTDs, residence time distributions, in that paper.
Okay.
And I could pull that up alongside if it's helpful to see that.
Yeah.
Or Anthony, do you want to pull that paper up if we need to reference it?
Sure. Actually, I can do that. Just give me a second to find it.
As a side note, we were a little cheeky in our naming of that paper, right? We titled it a very boring 120 hours, 15 million tablets under a continuous state of control. And it's turned out that's one of the highest cited papers I've ever had the pleasure of writing. So anyways, I was
wasn't too boring.
I think it was you. I think you were the one who chose the very boring aspect.
I was surprised the editors let us keep it. Yeah.
Yeah.
Anyways.
And the feeders, the 5 feeders on the left, they are all providing different things, or are they all? Can they be changed? Can you flex them around? Meaning, put different stuff in different feeders.
Yes. Yeah. So for this experiment, it was constant, but the technology in general, the reason we like this for continuous tableting is each one of those feeders could hold an active ingredient, a second active if you need, or an excipient to bulk up the material, provide different attributes for the drug product. And so in this case, they had consistent composition or I guess a single raw material in each feeder for the duration of the 120-hour campaign.
Mm-hmm.
But for example, we have this kind of technology at our plants in Ireland, and Anthony has in his plant in Portugal and New Jersey, and so the composition in each feeder will change based on the product being manufactured that week.
So are these things powders, or are they liquids, or is it some are powders, some are liquids?
This is all powder based.
It's all powder, including the lubricant in feeder 7.
Despite the name lubricant, which makes you think like oil, it's a solid lubricant. Magnesium stearate is what's present there. So it's anyways, it's a waxy substance if you put it in your hands. So it still has the lubricity to prevent like kind of sticking of mechanical parts, but it's solid in nature at room temperature.
I liken it to Crisco, although much solid, more solid form.
Chemically, it's pretty close to Crisco.
Yeah, exactly.
So if these are powders, how do you ensure that the density remains the same? Of course, it will fluctuate. That's one of the things with the tablets. But are the speeds of the machines, are they aligned so that we don't get air gaps in the powder, or the density doesn't become too much or too little as the stuff is moving through the pipeline?
Yeah, that's part of our development process, is to the density will fluctuate based on the flow rate through the system. But it doesn't change substantially over the ranges we studied. I mean, we studied during development anywhere from I think it was around 15 kilos per hour all the way up to about 100 kilos an hour, and over that roughly 6 or 7x kind of multiple flow rate ranges, the bulk density of the material is relatively consistent. But then part of the control process, like the bulk density becomes really important in the tablet press, where the powder has to flow into a die, and then a fill cam controls a bottom punch for the tablet press to basically pull in the right amount of material to achieve the target weight of the tablet upon the compression event. And basically the feedback control will adjust for that change in bulk density as the flow rate varies. So essentially, yeah, the bulk density will change, but the control system is designed to handle it as long as it's, not too massive.
Yeah, and that's the beauty of steady state too. So the actual bulk density that is dynamic in nature due to the process at steady state, it's the minute differences from between materials is relatively insignificant when you think of the blenders, right, and the feeders and the tablet press already have controls to manage it. So
there's another question.
Yeah. Yep.
May I?
Yeah, go ahead, please.
Yeah. I'm Fahime. I'm assistant professor in management science and information system in University of Massachusetts, Boston. So my background is far from mechanical and engineering background. I'm working more in operations management and data analysis, machine learning. But I found this topic very interesting because I think that from the data, if we analyze the data, we can get some insight in terms of the operations management or to improve the operations in total. But my question is that since I don't have any background in mechanical and chemical engineering, and I know that for this process, since it's a continuous process, I need to find out if I want to predict that something happens in T+3, so I need to know that the timing of each of these process. And this is beyond my expertise, but I saw the paper, the 150 hours, 120 hours boring paper, that you have the time here. I found the formula for the residence time distribution, because this is the one that's very important. And I found out that based on my knowledge, I found out that that paper, the process is the same as this paper.
Yes, that's right. It's just the data set.
Exactly the same. So I can use the RTD time that are in that paper for this project, for this data.
Exactly.
Okay, so I don't need to go through the mechanical parts, the chemical parts, those type of the equations. I have everything from that paper.
Correct. Yeah. I apologize if that wasn't clear. But to clarify for everyone on the call right now, it's the same experiment, the same data set. Our original paper was simply our own summary of that 120-hour run. And then, when I met Tucci and she convinced me that publishing the entire data set around that would be beneficial, we basically took that same set of data and just cleaned it up so it was better understood by folks that don't work in pharmaceutical manufacturing, and then just made it available and wrote this paper for your audience.
Okay. Thank you. Yeah, that's very helpful.
And,
all right,
and Bob, I'm new to all this. Is are the RTD profiles published too with the data set, or it's just qualitative from the 120-hour run?
No, we didn't publish any of our kind of derived results, Anthony. We just published the raw data. So basically the process parameters, as well as the kind of the quality results, right? The tablet, your tablet weights, that kind of thing.
Got it.
Sebastian?
Yeah, hi, thank you. Thank you for so I do have to leave soon, so I will make my question, which is very boring question, and it's about the data itself, like a data cleaning. And there are some variables that are some of the data in the live feeders one that's supposed to be positive, all of them, and there are a few instances that they are negative values. So that is one detail, and another detail is in some of the binary variables. For some of the columns, I could go explain the details, but in some of the columns, there are some values that are higher than 1, 14.5, 14.5 onwards for more than 5,000 rows. And in some cases, the number is, okay, so it's higher than 1, and in other cases, it's between 0 and 1. So how to treat these cleaning parts?
Okay.
Mhm.
So first on the loss and weight feeder one, can you just clarify which column of data you're referencing, or which variable?
Sure. If I can share my screen.
Sure.
Yeah, that'd be better.
Okay, sure.
You have to be a co-host.
No, I'm sharing now.
Okay.
Yeah. So, okay. So these ones are. This is a print screen of the columns of the rows that have issues. So, for example, this one is bold mod PD1. Here I have the timestamp, and the value, it is this one, 14.5, and this should be between 0 and 1, right? So as we can see, if I'm just moving down.
Is there zeros and ones in that column?
Yes,
Also?
in this column, yes. For some dates, there are for some dates they are zeros or ones. Yeah. There are some rows.
Sounds very peculiar. It feels like there might have been a column shift, because there were many. I'm not sure if I don't know, Bob, how this was uploaded, but if there's
Oh, these are fractional values.
if we might have downloaded during the time, we may have downloaded sections of the data because it was just so large. I'm not sure it all came as one file back then. But
Yeah. To build on what Anthony said, we expect a volumetric mode for PD4 or PDX. That should just be a binary variable, 0 to 1, 0 or 1, representing whether 0 is it means it has feedback control on the feeder. It's constantly measuring the weight and adjusting the screw speed to dispense the right amount of material. And then a value of 1 indicates it's volumetric mode, meaning that feedback control is off because some disturbance. And so it is not taking active measurements, and it's turning at a constant screw speed. And so, yeah, I'm a bit confused. I'm afraid Sebastian will have to dig in that a little bit.
Sure.
And
Let me send you this file or a summary of this file, both, and we can take it offline, perhaps. It could be helpful for
my
everyone.
bet would be, and I remember
Thanks.
working with this data, my bet would be that at times when putting the different files together, there may have been a column shift. You might, and I'm not sure how that data was saved or stored, or if it's one file from Jim. But that would be my guess.
I understand totally. Like exporting file from this large file from machines, or for anything in particular, large files is always there. These kind of things can happen.
right. Yeah. Thanks for bringing it to our attention, Sebastian.
Totally.
And that was, did you have two parts to your question, or did that get both, everything?
Oh, no, those were my questions. Yeah, very boring questions, very data questions. Thanks. Thank you, guys.
I mean, just following up on Sebastian's question, there's also noise sometimes where there's a huge fluctuation. Do you ignore it, or is this a physical issue with the machines, with the machine?
So the specifically the PD, the PD stands for powder dosing, or it's a loss and weight feeder. The way those work, right? So it's physically a hopper of material holding maybe a kilo or so of powder, and 2 screws sitting underneath that are turning to dispense at a constant rate, and then load cells underneath that measuring the differential change in mass as the powder leaves. And so it's inferring that any change in the mass over time, mass 2 minus mass 1 divided by delta time, that gives you your mass flow rate. But of course, if someone bumps into it, or there's a big vibration or something like that, that will disrupt that load cell. So
There's noise. Okay.
That noise will cause it to go from gravimetric mode into volumetric mode, and in that signal that we were just talking about with Sebastian. So yeah, sometimes that noise will show up in the signal, and then yeah, I mean, the best way to handle that, you can try to just ignore it and kind of filter it out, or you can average over a bigger time period, because like if it's a vibration, if somebody bumps it, a force will go up, but then it'll go down rapidly. And so if you average from over a long enough time span, it'll the positives and negatives in the noise will cancel each other out.
Can I add one contextual point to that? I assume the data you have is every second. Is that right? It's every second, Bob?
For that variable, yeah.
Meters? Yeah, because the system is actually monitoring data at a much higher frequency, right?
Yeah.
But it's the raw. It monitors the raw data, and it's not doing filtering it for that 1 second level. What it's doing is it's pulling a data at every 1 second, right? 1,000 milliseconds, it plucks the data out, whatever that is, from the raw data, from the real raw data, right? But the system only reports to us every 1 second. So there, just want to give that context.
Oh, okay.
Think so?
Yeah, thank you, professors and Mr. Miro and Mr. Tanducho. So I'm a PhD student in the Hong Kong University of Science and Technology, and I'm currently sort of quite interested in this problem and have been looking into it for a few weeks, I think. So I got a couple of questions as well, if you don't mind. Also, just to build on, I think Sebastian's question regarding I think he just showed the volume mode, that variable. I think there's also a similar problem in another variable called the refill actor. I think refill action. That variable is supposed to be binary as well, but it also shows some other like over than 1 or over than 0 values as well. So yeah, just regarding that question. So if you can help check that, it would be really appreciated. And also, my other question is regarding, sorry, let me check my notes.
Before we go on to your next question, I guess I knew off the top of my head which variable Sebastian was referring to, but could you further clarify? I'm checking the paper really quick.
Yeah, sure.
I don't know exactly which.
I can share my screen.
Probably the
as
refill
well.
act, right? It tells you when the refill is actuating.
Oh, the refills. Got it. Sorry.
Yeah, refill.
I would almost propose, like this again, like as Sebastian said, this is actually common when dealing with large data sets. You have to extract files and reassemble them. I would challenge, say, can you figure out what the right how to line up the data, right? I bet you my hypothesis is that there's a shift between one block of data and the next, and is there a lineup which might make sense, and then we can go, that would help us instead of digging through all the data to figure out if you could find one that's logical.
But some like we have different feeders, like 6 feeders, I think. For some feeders, those variables are normal, but for others, it's just a bit messy. Yeah, I think I think it's hard to align them align each other, So yeah, maybe we have to check the source, the data source, and see if there is some kind of shift.
Yeah, and to build on what Anthony said, yeah, I mean, you're correct. It's once a data set becomes too big to handle in Excel, like honestly, I don't have the skills to kind of inspect it visually like I would like to for this kind of thing. So if you see, I call it a frame shift mutation in the data set. If you see that, based on what you see on the other variables, like shifting everything over a column makes it make more sense, that's probably the correct answer. That's what I'd like to do. So once we have a chance to take a look at what Sebastian pointed out, what you're pointing out, we'll look for that kind of thing. But perhaps you have more sophisticated techniques that could help unearth these kind of things. Because yeah, in pharmaceuticals, like this is just not part of our training. I've worked in this business for 22 years, and we haven't. Not until the invention of continuous manufacturing did I ever have to worry about data sets that were anywhere close to the size. And
Yeah.
and it didn't go up just a little bit. It went up by like 5 orders of magnitude, like with 300 million data points when I was used to dealing with, 10,000 tops before that. So anyways.
One piece of advice, if this interested you, this is a normal problem to solve, is, at the point at where things obviously shift, if you broke up the data set between those two spots, what would the mean and distribution look like for each set of the data? And is there some kind of alignment or reshuffling, mutation, whatever it is, where the data just looks like it actually belongs here? And then I think that would be interesting. That would be the approach I would take if I had time.
Yeah. Thank you. Thank you for the suggestions. I think I also look into it from the perspective of shifting problem. Okay. So sorry. My another question is about
Yeah,
also
please.
about about the residence time distribution stuff. Just like Mr. Miro mentioned before. So we also noticed that there's a paper along with this. I think it's a whole series of work, and this another paper. I think one of the authors is Mr. Tantuccio in the paper titled "The Development and Use of Residence Time Distribution Model Control Strategy for Continuous Manufacturing Drug Product Pharmaceutical Process," and it's published in 2022. If you still remember that. So I think in that specific paper, it's also about building a residence time distribution model. There's I think you use a lot some techniques like NIR, some NIR data, some and not just and also some impulse experiments to build the RTD model. So in this specific research, this M&SOM 2024, there's also a research about this RTD modeling. I'm just curious about, because we obviously don't have access or any data access to the to the previous experiments done by you, because it's raw data. So should we invent this RTD model from a pure data-driven perspective, like without any other physical experiments? So I'm not sure if you can share some of your insights regarding this question, maybe. Yeah.
I think Bob, the mandate or the question was to find the RTD using the raw data itself, right? But in that, you can use the 120-hour paper, the boring 120 hours, as a reference, right? Because there are RTDs for the system, blender 1, blender 2, and I believe the tablet press in there, since that is the data set, right? So you can use it as a qualitative check, but you shouldn't need to do additional experiments or need additional data, I guess. It's a way to make sure you're not biased, I guess.
And
I don't know if that was answering your question, but
yeah. So a pure data-driven method to build this RTD may be preferable. I mean, in this specific question, right?
Yeah, and let me motivate that question a little bit more. So we were making acetaminophen, right? A very well-studied drug with lots of safety data, and so there wasn't much hazard with running these experiments and taking measurements like the residence time distributions that are shown here. But one of our nearly identical systems that I have at my plant in Ireland, Anthony has one. It has we often operate hazardous compounds or cancer drugs or things we haven't studied very well that we can't get in and use the same techniques to measure RTDs. And so having a non-invasive method to measure RTD is incredibly valuable because we simply can't open up the system in the same way that we did in these experiments. And so that's why we were really interested if RTD is a fingerprint of the process. That fingerprint is present across all the data. If we can just use signal processing techniques and other methods to unearth it, we could comply with FDA requirements to take these measurements, but in a hopefully more clever way that might allow us to also observe the kind of the safety requirements we need during operations.
Mm-hmm. Yeah.
So
definitely,
yeah, the raw data, we didn't publish the actual CSV files, but hopefully there's enough data within these graphs that you could get what you need.
Mm-hmm. Yeah. Thank you for answering. Yeah.
To check and ask, or sorry, go ahead, Mingze.
Mingze, do you have any other questions? I remember you had a long list of questions already.
Yeah. I actually have another one, but I mean, Mr. Sodi, you can go ahead, and I can wait later. Yeah, you can go ahead. Yeah.
Okay, quick one. Is there variation in quality of the basic feed? Those 5 feeds, is there variation in quality which would eventually affect the variation in the tablet quality?
Yes, that's part of the challenge for the control system and for the operations and for the engineering team during development. We have to choose the operational parameters that optimize quality and productivity. So we're trying to pump powders essentially, right? And pumping liquids is pretty easy. You can get very precise flow rate. Pumping powders, they're compressible, they're kind of inhomogeneous, and so there's some variability. So you'll see variation in the flow rate coming out of the feeders. And so one of the most fundamental quality attributes of a tablet is just the percent composition of the API and the other ingredients. And so the API, the active pharmaceutical ingredient.
Yeah.
And so our target might be, I guess, is 100%, or oftentimes 100 milligrams. And we typically tolerate anywhere from 95 to 105% and call that good quality. But so we can see, depending on the time scale of analysis, right, over second to second basis, you might see a fairly significant change in the loss in weight signal from a feeder. Over a 30-second time window, that fluctuation drops down. And of course, the control system is constantly monitoring this and trying to correct for it.
Okay, thank you. Then what is the unit of time? I know it just maybe sounds, because the system is monitoring things in milliseconds, we are getting data in 1 second or 30 seconds or 30 minutes even, depending on what we are measuring. But when you say we want to optimize the system, what do we think of as the unit of time? Or is that a well-formed question?
I mean, it might benefit from a little bit of clarification, but I'll say that our base unit of measurement, in my mind, we often think in terms of the residence time in the blender.
Okay.
Any variation in the feeders that happens on a time scale shorter than the residence time in the blender should get blurred out by the blender, right? The blender is there to dampen out any fluctuations. And so the residence time is on the order of a minute. And so that's why we tend to think about like, a 30-second averaging of the feeders. That's as long as you're kind of in a state of control over that time span, then the blender will dampen out that fluctuation and maintain the system in an overall state of control. And so, yeah, we tend to think of it in terms of the residence time distribution.
Okay. And then my final question is about the output. On the output side, this is where the tablets are coming out. So there's tablets, are they somehow visually or some other way quickly separated into what tablets are acceptable, what are not? But then you still do the API control check, but that's in a way not part of the process. Meaning it's not part of, it's not taking stuff out from this continuous process. If you show the output, the other paper back, the main paper and the diagram.
Can I say one thing here? So I
Yeah,
think,
go for it.
you know, when producing tablets, I'm not
Yeah.
sure the details
Yeah,
of the
this one.
data set here, right? But you're taking tablets as samples at line, meaning they're not,
Yeah,
it's just taking a sample from the flow, a very small sample at some
yeah,
frequency, right? And that does not impact very much the yield,
yeah.
how much material,
Okay,
right?
yeah,
But then there's tablets that, you know, the rest of the tablets are going through the process of being collected in the bulk,
yeah,
right? And you have information on those tablets sampled from the stream, right? And you have information from the machine, like those tablets that are sampled stream, you have actual tablet measurements, tablet weight, hardness, thickness, diameter, maybe the NIR or the spectra of the content. And then the inline tablets, you don't have that data, but it should be similar to the data that we pulled, right? For those tablet attributes. But you do have all of the very more higher frequency machine data, right, around the tablet press, which made those tablets.
Yeah.
Right. And so the
Yeah,
challenge is, how do you match those two up, right, in time, and also, you know, quality, or inferring quality of those higher frequency tablets when all you have is the real quality of the sample tablets.
yeah. To build on Anthony's point, there are two control loops active at the same time. I'll just call them the fast loop and the slow loop. So the tablet press is, you know, its goal is to make as quickly as possible tablets of uniform weight. And you know, it's bringing two tools together to compress powder in a die, and it's measuring the force. It's measuring a lot of things, but primary variable here is the maximum compression force as those two metal punches come together and squeeze that tablet in the die. And so it's using that force data to quickly decide, am I within a control, a state of control within the target? And then it will decide based on that, does this go to the good tablet channel or the bad rejection
okay.
tablet? And then in addition to that, every, you know, 10 minutes or so, the at-line checks that Anthony described, the tablet analyzer measures the
Yeah,
weight, thickness, and hardness to make sure the force control loop is still doing its job. And if it says, oh, you're drifting a little bit, it will send a signal back to the tablet press to adjust that fast control loop.
okay, yeah, thank you.
Yeah, thank you. So I also have another question. So sorry if I take up too much time. So
We are here to
my-
answer questions. Go for it.
Thank you. So my another question is also about let me check these notes about the very boring 120 hours that paper. So I think it's very quite informative. I think along with this M&SOM driven paper at
Mingse, I can't hear you.
Yeah, we're losing him.
thing screen. So
Oh,
so here we are in the 120 hours that paper. So my specific question is about the review times here. So we can see here, for example, we will take PD1, for example, here. We can see there's like thousands of review events, but in the log data given by the data-driven challenge, like if we can get PD1, sorry, we can get so here is PD1. We can only get like less than 100, like only 40 around events. So could you, I mean, maybe there's some domain knowledge that we are not that familiar with. So I'm not sure if you could share some of your
No,
insights on this. Yeah.
I see what the challenge is. This is more a nomenclature problem, but let me attempt to explain. So what's not shown in the diagram, there's actually two different refills that are referenced, and I think we call them both refills, but there's two different refilling systems. So there's essentially, if you imagine our continuous manufacturing system, we had big drums of like 50 kilos of powder for all our different raw materials. And those get transferred into a tablet press. Yeah, thank you. That's the CDC 50 unit that we use. So those big drums get transferred into, if you look in the center of above the white flexible elements, that's a pneumatic transfer station which sucks powder in from the drums and holds it above the loss and weight feeders, which are below those little white flexible units there. And so, yes, that's the loss and weight feeder. So, and you can see the pneumatic receiving unit above, right? So that white thing in the middle is a ball valve, or we call it the top-up valve. And so I think in the one data set,
Are you referring to this part? It seems a bit laggy. Yeah, it's all right now. Yeah, sorry, just a bit laggy before. Yeah.
Yeah, sorry. So in the one Excel file that you showed briefly, Mingji, we were describing the refilling of raw material drums. And that's like, there's a small number of these, 50 kilo drums you can see there of Avicel 102, microcrystalline cellulose. And then so that drum of 50 kilos each, that gets pneumatically transported into that receiving area, and then where that white ball valve was that you showed in the other diagram, that white ball valve actuated 2,700 times, and to top up the loss and weight feeder that sits below it.
So this kind of column of data is for this one?
Exactly. Yeah.
So, and in the paper, there's many events, and these events are for the inside refilling, right?
Yes.
Thank you for clarifying this.
Yeah, and sorry for using the same term to refer to two different things.
It's okay. So also my last question is about this one. So, sorry. I think this also is about the raw material refilling stuff. So in this MSD data-driven challenge paper, they also propose a question regarding like if we can try to detect the time of the raw material change time. So from that perspective, I think this refilling time data is kind of very important. So could you share some insights on how this estimated refill time and this data or time, the difference between these two variables, like how they are recorded, or what's some kind of methodology behind these two columns? I mean, because I didn't see any references of their methodologies in either paper. I mean, as of now. Yeah.
And Anthony, do you want to try to field that one?
I'm looking at the data. I'm wondering if my first thought was that it was just a column typo, but it seems that there is a date time and an estimated daytime for each material. And they're different. Why would that be?
This is a trip down memory lane, Mingji.
So
I think I'm gonna guess here, right? You know, it may be that, you know, that, yeah, no, I'm not gonna guess. Forget it. I'll just be
So
confusing.
while Bob and Anthony are taking a closer look at it, because even a few minutes might refresh their memory, but looks like Fahime has another question. And, you know, because I don't mean to cut you off, Mingzi, but just looking at the clock, want to make sure that Fahime and maybe Shanshan or somebody else might have questions too. But in the meantime, they might think about what might be going on over here.
Thank you. Thank you. Thank you for sharing this meeting. Thank you.
So I have one other question. I was wondering if the data in the MSOM paper, does that data include the spike study that you consider in the other paper? Because in the other paper, 120 hours paper that you study the residual times, residence time, there are spike study data too. So if we want to use that time in that paper, do we need to have that data, or that data is available?
So we'll have to dig into the data in more detail to probably answer that fully, but my recollection is that we you know, the spike studies are very fast. We basically get a little weigh boat of the active ingredient, and we just pour it into the system really quick while everything's running, and then we, you know, wait a couple minutes to measure the response. So my recollection is, you know, the system was running the whole time, and we just did that quick weighment and then that measurement of the response. So I believe that data is part of the overall data set. And so we'd have to go and look and see exactly when those few events happened. But we didn't exclude any data from that overall 120 hours in what we provided in the MSOM's paper. So, but because that you know, the system is nearly entirely automated except for that little we call it a spike, right? That weighment of active ingredient. So that how much we poured in, that is not represented in the CSV files that we provided as part of the MSOM's paper. So, you know, it's, for the most part, the data is there, but because that was done manually instead of through one of the mechanical systems, that one little addition to the system was not included. Does that make sense?
Yeah. Okay, thank you.
Yeah.
Hi, can I ask a question? Yeah, thank you, professors. Thank you for organizing this Q&A session. So my name is Shanshan. I'm currently a PhD student in University of British Columbia. So I have several questions. So first, I want to ask something about the sampling strategy. So, I mean, can I share my screen?
Yeah.
Yeah, okay. So can you see my screen now?
Yes.
Yeah, so I think in Horman's reference, so they explain that, so they will sample the tablets in like 2 ways. So one is like every 15 minutes and one is every 30 minutes. So I just want to know, so for like these 10 tablets, so are they just generated at that moment or not? I mean, for example, if I want to like sample 10 tablets at 8:30, so will these 10 tablets are generated at that moment, or they are just generated from like 8 o'clock to like 8:30?
No, we were fairly precise in our you know, taking that sample at exactly the moment that was described.
Okay, so then I have another question. So if I want to build some predictive model to predictive the quality of the tablet from a series of like machine parameters or machine data set. So since I only have the information of the tablet at that moment, I mean at 8:30, so how can I like adjust these machine parameters? Because it seems that I can also, sorry, go ahead.
Well, I think that the short answer is through the residence time distribution that we were talking about earlier in this discussion, you can time shift the time series data to say, what was dispensed from the loss and weight feeders at, let's say 8:25. If there's a 5-minute residence time mean, then it will be leaving the tablet press at 8:30. And so that frame shifting in the data will account for the, what you measure at 8:30. And so we were, proposing that the MSOMs community try to test different methods for that frame shifting in order to make predictions.
Okay, yeah, okay, I see. Yeah, thank you. And my another question is about these optimal release policies. So here I noticed that you raised the concept of release by exception. So can I know like more, can you explain it more? So I feel a little bit confused about this concept.
Sure. And this is a very popular concept in pharmaceuticals, but it's never been achieved. So I would love insights from really, you all have experience in a vast number of industries, whereas I've only ever worked in pharma. But the general concept is we kind of do a retrospective analysis of every of the batches we make, and we kind of test a bunch of tablets or a bunch of drug product against a whole array of measurements, critical quality attributes to say, is it within the proven acceptable range for each of these critical quality attributes? But an alternate method, one might say, as long as I know my process is in a state of control and I've got a lot of knowledge around the process and it's the way it's controlled, maybe as long as all the process parameters stay within, a narrow window for the duration of operations, perhaps I don't need to test any tablets at all because I know everything produced within that narrow window is going to produce tablets that meet our CQAs. And so that's what we call release by exception. That the exception, doing release testing, is done only in the exception where the process was not controlled within that narrow window. If it stays within the window, you don't need to do release testing. If it goes outside of the window, you do release testing because it was an exceptional event.
Okay, yeah, I see. I think that's a really like interesting idea. Yeah. And also, sorry, I have a last question. So it's about for this process flow. So I noticed that we will only like dispose the bad tablets in the end, right? So may I know, is it possible if we like divert some raw material in these intermediate steps, if they does not satisfy the quality requirements, can we divert some materials in the intermediate steps? Is it possible to do that?
Not in the way the system is currently configured, but certainly if you were to, make mechanical adjustment.
Okay.
But as the system's currently configured, it's only possible to do rejection either after the tablet press or after the film coater. And to be clear, before the tablet press, all you have is powder, and so it's actually kind of messy, right?
Yeah.
To do an inversion, you'd be kind of diverting bulk powder, which is fluffy and kind of imprecise in terms of its properties.
So you mean like diverting the powder in the intermediate steps is maybe a little bit noisy, so it's not like workable. Okay.
I'm not
It
seeing
would be
in
difficult to
continuous
do.
flow, right, Bob? Sometimes if people if processes discretize it into powder like granules, right? Sometimes you send plugs
That's
of
true.
granules. That's the only way you would divert it. But this system is not set up for that. And
Okay, yeah, I see. Yeah, thanks, Q, for your explanations. Yeah, that's really helpful. Thank you.
I can.
Yes, go ahead, Mohan.
Oh, yeah, this is a sort of a strategic question as far as the competition is concerned. There is a lot of data. Is it fair to say that what I'm just paraphrasing you, that the goal here is to figure out how to optimize the system so that we can get the best quantity and quality?
Yeah. Yeah. I mean, this continuous manufacturing, from my perspective, it's true that it's very new to the pharmaceutical industry. I recognize that, petrochemicals has been doing this for 100 years. Food industry has been doing it for at least, probably 75 years. But pharma, we've been doing it like, I don't know, 10 or 15. And we're still trying to optimize how can we comply with regulations? How can we make high quality products? But also how can we, lower costs and drive efficiencies that allow us to, get our medicines to more people quicker, faster, cheaper, So I think that the what you pose there, Mohan, is spot on, and there's lots of ways. I truly believe that we can use data to do better than we've done in the past, because right now we're applying all the old methodologies we had for releasing product, producing drugs. We're applying those old methodologies to this new way of manufacturing, and we need to make those adjustments. And so we're kind of asking the MSOM community, hey, can you come up with some ideas? Some maybe it's just transplanting an idea from another industry and saying, why don't you do in pharma, why don't you do the same as they do in this other industry? That kind of thing. I'm a bit naive on that kind of.
So wouldn't the result be some kind of a trade-off curve? For example, I could run the system really fast, but then many of the tablets I get will have to be trashed.
Absolutely.
Or I could run it really slow. I may get very high quality input, but it's just will be too little. So are you looking for a trade-off curve of some kind, or is there a one optimal solution?
No, that trade-off curve is exactly the kind of thing that I think would be helpful, and it's something we don't often do. We kind of estimate it mentally, but we don't take a data-driven approach there. And make you can use our data to produce one that operations management within my company can make decisions every day on.
Yeah, it will be really cool to build a trade-off curve. That will be really useful.
I'm still thinking, compared to chemical industry or even auto manufacturing, you've got a bigger problem because you've got powder. Powder is, I've seen powder in chemical industry, but there the quality did not really matter, like fertilizer. It's also powder, but I mean, if the quality is this or that, nobody really cares, but you're producing medicine. So it is a tough problem. Okay.
Well, thank you.
Thank you.
Thank you, Tuche, but huge thanks to Bob and Anthony, and really exciting, all the detailed questions that everybody had over here. So good luck, guys. This is very, it's very nice to see this.
Yeah. Thank you so much.
Thank you so much
Thanks again for having us.
Bye, guys.
Yep. Have a good one. Bye-bye.
Thank you.
Bye.